We turn now to the presentation from Justin Ichida, who is the John Douglas French Foundation Endowed Associate Professor of Stem Cell Biology and Regenerative Medicine at the Keck School of Medicine at the University of USC. He is basically someone we've all known for many years, who trained at Harvard with Kevin Egan and also Jack Szostak, and did incredible work on cellular reprogramming, which has already led to some striking reports and findings related to reproduction of features of sporadic ALS in iPSC-derived motor neurons. His talk today is using iPSCs to model specific sporadic ALS and to evaluate therapeutic strategies. Thank you, Justin.
Well, thank you, Bob, and to the organizing team for the invitation. I've really enjoyed these presentations so far, and looking forward to the rest of them. And happy to participate in this, this session on sporadic ALS, because it's something that we've really been kind of focusing on turning towards in the last few years. And of course, because it's a part of ALS with such a high unmet need. And we kind of just think of this in a cartoonish sense, where there are different drivers, genetic drivers of ALS. And, in thinking about how to identify therapeutic strategies for the sporadic sector, of course, the challenge is that for most of those patients, we don't really know the exact composition of the genetic, drivers of their disease. And technically, some of them may not have strong genetic drivers. And so that just makes it difficult to generate, laboratory models of those forms of disease, and therefore test, agents, test articles before they go into the clinic. So that's kind of the gap that we were trying to fill. And we basically hypothesized, a few years ago that, well, it's, if many of those sporadic ALS patients, do harbor genetic lesions that contribute to their motor neuron disease, then if we were to generate iPSCs and motor neurons from them, then their motor neurons may exhibit some of the features of ALS, disease pathogenesis, similar to those from familial ALS patients, with drivers like C9orf72. But this was really not clear if this would work. I mean, first of all, you were talking about a very young, sort of immature neuronal culture, trying to model a late onset disease. And of course, we still didn't really know how many of the sporadic ALS patients' cells might actually have genetic causes of motor neuron disease. And so nevertheless, we forged ahead and tried to see if this was possible. And so again, just, we started with kind of the C9 population. We generated, induced motor neurons from iPSCs from many controls, about 9 controls here, and about 9 C9orf72 ALS FTD donors. And I should say that, many of these, a good portion of these lines were from Answer ALS. And so I really want to thank the patients and the investigators who participated in that, pretty, wonderful initiative. But you could see that the rate of death, if you just track these neurons in culture, the rate of death for the C9 lines was actually quite elevated compared to the average of the controls. And then we could also see here that if we looked at the TDP43 localization in the neurons, compared to the controls, which were predominantly nuclear localized TDP43, we could readily see some of these TDP43 puncta out here in the cytoplasm of the C9 neurons in culture. And then we turn to, looking at the sporadic ALS lines. And then, to my surprise, these are all the sporadic lines we looked at, and they essentially all had higher rates of neuron death in culture than the controls. And so that, I think, and I would also say that if you look at their TDP-43 localization, you can see that a lot of them, like this neuron here, had, a lot of localization out there in the cytoplasm rather than in the nucleus. So I think, there could be multiple explanations for this, but I think one simple interpretation is just that, indeed, maybe many of these sporadic ALS cases, patients harbor some genetic drivers of motor neuron disease, and of course, they could be their ultimate, onset of ALS could be induced in addition by, environmental factors and other things. But I think it does maybe suggest that these sporadic ALS neurons could be useful for identifying, agents that could mitigate, the cause of death or the TDP-43 mislocalization. And so that's kind of how we're using it now. And so we then turned, towards trying to ask if we could identify, modifiers of disease progression in these cultures that might be, somewhere of a convergence point between a lot of the sporadic and unknown cases, as well as some of the familial cases. And the one thing that, it was relatively obvious, and I think has a decent amount of, experimental support as well as genetic support, is that TDP-43 pathology seems to be common to many of these cases. And so in order to identify some new targets that might be able to modify TDP-43 pathology in the hopes that it might be able to address sporadic ALS, we turn to, looking at TDP-43 mutation lines. And of course, the thought out there, I think there's a good amount of evidence now building that, a lot of the mutations that occur in TDP-43 and ALS or FTD patients occur in this sort of disordered region at the C-terminus that kind of increases the propensity of TDP-43 when in relatively high concentration to form kind of irreversible aggregates. And so that, this could also be caused without the TDP-43 mutations if the concentration is higher. But ultimately, I think a similar type of aggregation occurs. And so, this it suggests that, maybe a line with the mutation in TDP-43 might ultimately mimic some sort of pathology that's like, most of the ALS cases, which just have normal TDP-43. And so Jasper, Ruben Sigler, and Sharon too have been really leading this effort in the lab. And we were fortunate to be able to work with Ken Kosick and Juliana Acosta Uribe from UC Santa Barbara. They have been traveling to Colombia for many years and think they've identified what, one of the largest families of TDP-43 mutation carriers. They range from, people with ALS and also FTD or a combination of the two. But they all have this I383V mutation. And so we actually generated, iPSCs and motor neurons from one of those patients with that mutation. And so Jasper's method is really to generate these induced neurons and then cluster them into a three-dimensional sphere, which he can then culture for many weeks quite easily. And one of the nice things about going for 4 to 5 weeks is that you really start to develop some of the TDP-43 pathology. And so you can see here that if he generated induced neuron spheroids from C9 patients, just like kind of the previous slides I showed, you get a faster decline in survival compared to controls. And the same was true of this patient with the I383V mutation in TDP-43, a fast decline compared to the controls. And so given that was working, Jasper decided to use that kind of survival phenotype as the basis for a CRISPR interference screen where he could ask, if we suppress every gene in the genome in one cell or another, which of those gene suppressions leads to an increased survival of the TDP-43 patient neurons? And therefore, the nice thing about this kind of thing is that a gene found in this type of screen, with the advances in RNA sort of ASO and siRNA biology and drug development could be relatively rapidly translated into a compound that could be then tested, in patients. And so when he's basically recovered all of the neurons that were left, he identified that several, CRISPR guide RNAs suppressing different genes came up as enriched. Some of them we've already seen before, like PIC5 or the SIF2 complex, some of the nuclear porin genes that have been also identified. And then we saw one called VAV2, which we hadn't seen before. And what was interesting when we cross-referenced that to some of the newer genetic studies that had been coming out, we came across a study by Sai Zhang and John Kupernock and Michael Snyder, where they had essentially, tried to leverage the fact that, motor neurons seem to be in an, harbor or, selectively vulnerable in ALS, and it could be said that, mutations that are expressed in motor neurons are most likely to be a cause of the disease. And so they thought that they could increase the power of their genetic power by using some of the modern tools to only focus on, genes in the motor neuron transcriptome or in gene regulatory regions controlling those genes that are in the motor neuron transcriptome. And so they were looking then for kind of rare variants that had a, relatively large effect on the on the propensity to develop ALS. And so they did find actually in that study that actually their top gene that had rare loss of function mutations that were enriched in controls over ALS patients, and this comes from, a lot from the Project Mind database, was VAV2. So, and then on top of that, when they looked at the Answer ALS database, where they had essentially generated iPSC motor neurons from different patients, they found that the patients that had the latest age of onset also had like the lowest amount of VAV2 expression in those neurons, potentially suggesting that, some of the genetic architecture that influences VAV2 expression was such that if they had lower VAV2 expression, these patients lived longer. And then we asked, what happens in postmortem tissue to VAV2 expression. And so we looked at the Eddie Lee data that many people have now sort of been looking at. They had basically taken FTD or FTD ALS cortices and sorted out neurons that either had high or low TDP-43. And we found that the neurons that were TDP-43 negative or low had actually very low expression of VAV2. And this didn't seem to be caused by a loss of TDP-43 itself, because in the paper by Joe Klim, when they knocked down TDP-43 in iPSC neurons, that didn't have any effect on VAV2 expression. So I think one, possibly generous interpretation of this result could be that, the neurons that had lost TDP-43 that lived the longest in these patients actually had very low expression of VAV2. So all of these kind of converging around maybe lowering VAV2 could have a protective effect against ALS. And so indeed, we generated antisense oligos against VAV2. We could suppress it by about 50% in vitro. And then in either C9 or 72 neurons or sporadic ALS neurons or TDP-43 neurons, we could basically return the rate of death back to the level of controls by suppressing VAV2. And Jasper also found that while patient neurons have a lot of cytoplasmic TDP-43, suppression of VAV2 can, essentially reverse that back to what it looks like in controls. So a lot of nuclear localization. You can see the nuclear to cytoplasmic ratio quite dramatically increasing with the VAV2 suppression. He was also able to look at some of the cryptic splicing that occurs when TDP-43 function is lost. And so again, suppressing VAV2 with the ASO, you can see that this elevated truncated Stathman2 cryptic splicing occurs in the sporadic patient neurons, but you mitigate that somewhat with this VAV2 suppression, which really reflects, I think, the increased localization of TDP-43 in the nucleus, where it's presumably doing this type of work. And then, of course, he turned to some in vivo models. And so one of the models we use is a TAR44 mouse, which is an overexpression of human TDP-43. And here he's suppressing VAV2 by about 50%. You see quite a dramatic reduction in the gait impairment here, kind of a preservation of the motor neurons in the spinal cord, and then an increase in the survival of these mice, with a few of them living, quite a long time. So that's kind of one, new target that seems to be interesting. And I'm not going to go into the mechanism, but what's interesting about it, we think, is that VAV2 is an activator of these GTPases, and these GTPases actually, different ones actually repress different parts of protein clearance. And so by reducing the activation of these, we think we're actually, through a single target, activating a multi-pronged kind of pathway that could activate both areas of the protein clearance, autophagy and the proteasome, leading to neuroprotection. And then just for a couple slides, I wanted to illustrate kind of one additional way, I think, I guess, a direction we're moving into more. And this is in collaboration with Sai Zhang and John Cooper Knock, who I'll get to what they've done anew in a second. But another gene we found in this same screen was a gene called CCDC146. Had never heard of it before, but Sai and John had extended their previous study to now looking at variants that have a strong correlation with survival of ALS patients, which I think is also important to look at in addition to just risk of ALS. And they found that this genetic variant, which actually increases CCDC146 expression, it's right next to CCDC146, and increases its expression in motor neurons in vitro. You can see here that ALS patients that had that variant have a very rapid rate of decline. So their survival is very short. And so we thought that would mean that maybe suppressing this gene could be protective. And again, when we tested that in the C9 and sporadic ALS lines, we could reduce their degeneration. And then we've also been testing it in the same mouse model. And you can see that there's quite a big increase in the survival time of those animals. So, in conclusion, I think the important thing to remember is just that, at least in our hands, what we can see is that a lot of the sporadic ALS neurons from those patients really display, ALS pathology and rapid neurodegeneration in the dish, suggesting that, some of these genetic drivers are present and driving this difference from the controls. Through a CRISPR screen on a TDP-43 patient line, we've uncovered this VAV2 and a CCDC146 potential, new targets as being highly neuroprotective. There's some interesting clinical and genetic data that's beginning to suggest that, lowering these products could be protective either against risk of the disease or progression. And then, this is what it's looking, it is looking promising, I think, in patient neurons and mouse models. So I'd really like to thank Jasper and Sharon, who really did the lion's share of that work, in collaboration with Sai and John Cooper Knock. And then Michelle, Gabriel, Yichen, and Wen from Acuristem are really the team that put together that patient panel of lines that I showed in the beginning. So thanks very much.
Thank you so much, Justin. That's extraordinary work in general, but also incredibly exciting in terms of the recent work you've just delineated. Did I understand you to say that the impact of VAV2 and CCDC146 is evident in sporadic IMNs as well as those with several different ALS mutations? In other words, these are broadly impactful, kind of across the full spectrum.
Yeah. So far in the model systems, in the induced neurons, yeah, they work well both in the sporadic lines as well as the familial lines.
Is there any sense that there could be adverse effects of over suppressing either one of these? In other words, if
Yeah.
one looks, for example, an obvious experiment at a knockout mouse, are they
Yeah.
viable? Is there a dosing that's very important as you contemplate potential
Great,
clinical
yes,
patients?
great question. For CCDC 146, actually for both of these, there have been knockout animals that have been generated, and of course we've looked at sort of the genome AD, the Nomad database for mutations in the healthy population that might be depleted. But generally for CCDC 146, the knockout mice are normal, except the males have some infertility. So I think, there you might want to consider, like an ASO strategy where you wouldn't likely reach that area. And then also for VAV2, I think, the heterozygotes are fine. Knockouts can have some issues in cardiovascular issues. So there again, you might want to have a modality that's restricted predominantly to the nervous system. And I think there, the oligonucleotides could be a good, opportunity. Yeah.
Thank you. We have a question from Melanie Leitner, who asks, how can we reconcile the widespread death across the sporadic iPSC motor neuron lines with the multiple hit hypotheses that Amar presented?
Yeah,
Yeah.
yes.
Thank
I
you.
think that's a great question, and I welcome Amar to comment. But I think that, it's possible. Of course, I think the other hits can really influence the likelihood of developing the disease in a time where people are still around. So I think, that's almost certainly a big part of, who ultimately develops the disease. But I think I think it was surprising to us, I think, that we could see, phenotypes in the motor neurons of almost all these sporadic ALS patients that were not distinguishable to us in the dish from, C9 cases or, some of the other, familial cases that we looked at. Yeah.
Thank you. A couple more questions in the chat. Daniel Ori asks for the CRISPR screen. This is interesting. Do you know the UNC13 risk factor genotype of the patients in the study?
Yeah. I don't think we actually checked. Well, we do have, no, they are not I just remember that they're not the risk. So they're at least not homozygous risk, but they might be, yeah, that person might have had either non-risk or heterozygosity at that locus. Yeah. Not 100% sure.
And then the question is asked by Chuyun Chung, how common is the CCDC 146 SNP patient in the population, and particularly in ALS versus controls?
Yeah, it's pretty rare. So I think it was roughly about 10% of the patients had the SNP that would increase CCDC 146 expression. So so that, I think maybe you might, if we were to test this clinically, those patients might benefit the most, but you could, it would be interesting to see if other patients could also benefit just from general lowering of the gene. Yeah.
Thank you. Well, we're almost on time, so I think we'll move ahead and save subsequent questions for the panel discussion.